Hi. So my friend and I had this argument, and so I think it needs to be resolved. Let's talk about cell membranes and how materials go in and out of cell membranes, and that's the topic for today. Okay. Now, when we think of a cell, we think of it as a circular thing. It's not always that case, but that's okay. You can always go with that. Now, the cell membrane, whether it's plant cell, animal cell, doesn't matter, but we're going to stick with animal cells. We're going to talk about nerve cells primarily. Have cellular material inside which to stop it from dissipating. Basically, if cells dissipate, you're dead. Okay, so to keep cells intact, you have the cell membrane. Now, the cell membrane is a gate. It's a fence, and it is the perimeter that bounds all the material and keeps it inside. But for cells to function, you need material to go in and out of the cells. Now, I'm going to specifically focus on the ions that need to go in and out of a cell to keep it properly polarized. Now, why do cells have to be polarized? Nerve cells especially have to be polarized so that they can conduct signals and we can actually think and speak and function. Okay, so for nerve cells to conduct electricity, they need, it's not actually electricity, but yeah, okay, for them to conduct electricity, they need to be polarized in the correct way. You have to have a higher potential somewhere and a lower potential somewhere, and then the electric, the current has to flow. It's not exactly current, but okay, the current has to flow, and that's how the neuron sort of passes the message from one part to the other. Okay, so the importance of potentials being just the right level at the right place, not too much somewhere, not too little somewhere, etc., is very important. And how is that maintained? That is maintained by this very micro thing that happens, which is on the cell membrane. There is this side of the cell membrane, and this is this side of the cell membrane. So let's say I am the cell, and I am bounded by the cell membrane. Outside of me is the extracellular environment. Outside the cell, the environment, which is maybe blood, maybe plasma, or whatever. Okay. Inside the cell is cytoplasm, mitochondria, Golgi bodies, ribosomes, nucleus, all the good stuff, the proteins that the cell needs to operate. Now, because the cell membrane bounds the contents of the cell, it is completely doesn't allow any movement. But ions have to go, the water that you drink, the salt that you eat needs to go inside the cell. Yeah. So how does that happen? That happens through what is called ion channels. They're called ion channels. Ion channels. Okay. Ion channels are basically what permit the movement of ions like Na plus, K plus, Cl minus, potassium, sodium, potassium, chloride ions to move in and out of cells, calcium ions also to move in and out of the cells. This is important, okay? Now, how does this happen? This happens, now when you have a fence, how do you enter? Either there needs to be a gap in the fence, or you need to have a gate that opens for you, okay? A gate that is shut is no good. A gate that actually opens is useful. A gap in the fence doesn't need permission. You just go in and out. So the gap in the cell is called a leaky ion channel. So cells also have those. There are gaps on the cell membrane. Those are called leaky ion channels. This is typically how your sodium, potassium, they often leak in and out of cells. Okay. These are called non-gated ion channels. Non-gated ion. There's no gate. It's just open. Then there is the ion channels where there's a channel. Channel is an opening in the fence, which is basically blocked. But when a specific molecule comes and sits on it, then it opens. Okay, so this is like, your biometric doors where it's closed and your friend goes in and that says it doesn't open. You go and touch it. It says, okay, you are a valid employee, and it lets you in. So it's like that. So that when the correct ligand or the molecule goes into goes and touches that channel, it opens up. Examples of ligands are neurotransmitters, hormones, etc. So, for example, acetylcholine goes and it touches this little ion channel on the dendrite of the neuron. At the end of the neuron, there's this little cell, and on that cell, there's a little gate, and on that gate, there's a little biometric sensor, and the acetylcholine molecule goes and sits on it and says, hey, it's me. And the ion channel says, hey, I recognize you, and it opens, and the acetylcholine comes in. Okay, so that's how it works. Acetylcholine is a neurotransmitter. Okay, so that's the second type. So we had non-gated ion channels, and then now we had ligand-gated ion channels. Ligand, ligand, I don't have any space here. Ligand-gated ion channels. Then there is the third type. Third type, mechanoreceptive ion channels. Now look, nerve cells inside, it's okay, there are neurotransmitters and so on, so that's okay. But on your skin, sensory receptors, there is no acetylcholine falling from the sky, so it has to be something else. What we respond to is touch. And touch is two things, pressure and vibration. Think about this. If I touch, of course, I can perceive it. But if I even don't touch, but I wave my hand, I can feel the change in air, or the fluctuation in air, the fluttering of air on my skin. Why? Because the receptors on the skin, okay, there are 4 different types of receptors, sensory receptors, and 2 of them respond to pressure and 2 of them respond to touch, and respond to frequency. And the ones that respond to frequency, one is a high frequency detector and the other is a response to low frequencies. So the one that is on your skin is a low frequency receptor. Okay, so these then open the channel when they perceive this fluttering, and then the ion goes in, and then polarizes the cell, and then there is an action potential or a movement of signal through the neuron. Okay, then the last one, and that is a voltage-gated cell. So the first ligand was a chemical-gated cell, the second was a mechano-gated cell, and the third is an electric-gated cell, a voltage-gated cell. Voltage-gated cell, now see, normally neurons don't touch each other. Okay, when this neuron has to talk to this neuron, there's a gap, and it releases these neurotransmitters into this gap called the synapse. The neurotransmitters move to this side and do this biometric thing, and then they go into this neuron. Okay, so they are actually not touching each other. That is chemical gating, ligand gating. But in some specialized neurons, what happens is that the neurons are touching each other. It's a lot more efficient. And the potential difference between this and this directly causes the gate to open and the signal passes. Okay, the ions pass rather. So that is what you call a voltage-gated cell. So to conclude the argument, whether there is a difference in a chemical-gated cell and a voltage-gated cell, I think it should be obvious by now. Well, thank you for listening, and I hope you enjoyed this.